Okay, we're going to try this again. This is for the immune system, review for the test. Okay, what I'm going to do is I'm going to go through these slides very quickly, just so that you get them in the 15 minutes. And then I'll touch on things that, if I think you need to know it, I'll just go from there. So we're going to go try to get this whole chapter 15 slide thing in under 15 minutes. First of all, immune system. Chapter 15, defense mechanisms, skin, liver. First of all, you have innate and adaptive. You need to know that. The two categories of immune mechanisms in the body. Innate is inherited, and you have epithelial, stomach, liver, phagocytes, pathogens, but the skin, that's part of your innate immunity. Know what innate immunity is. Know what a pathogen-associated molecular pattern is, and that it helps distinguish cells. Tell the immune system that cells are not self, and that they might that they're actually foreign. Complement is part of the immune system, parts 1 through 9. 1 is activation, or I'm sorry, 1 is recognition, 2, 3, 4 are activation, 5 through 9 attach to cells and cause lysis of the cells. 3 types of phagocytic cells, neutrophils, mononuclear, macrophages, and in some organs like the lymph nodes, lungs, brain, liver, they have fixed phagocytes like Kuffner cells. Know that white cells can get out into the tissue. It's called extravasation. They are also attracted by cytokines. It's a chemical attraction in the lymphs. It's called a lymphokine. Cytokines will attract other white cells. Lymphokines will attract lymphocytes. Phagocytosis, we've talked about. Know it. Fever by the hypothalamus. It's caused by pyrogens. Tylenol is antipyrogenic, so it knocks down fever. Interferons are produced by cells that are infected by viruses, help cells that are not infected fight the virus. Adaptive immunity is acquired. You have to be exposed to the pathogen, i.e. Measles or mumps, and then your body will make an antibody against it. Antigens are anything that cause an antibody to be formed. Molecules have different sites that the immune system recognizes as foreign. Those are antigenic determinant sites. Haptens are molecules that antigens can be bound to, make them more effective, especially in creating vaccines, make them more antigenic or make the body more apt to make the antibodies against it. Immunoassays we use, I'm not going into that again. Lymphocytes from stem cells, you have 2. You have B and T. T tend to fight viral or fungal diseases. B are going for bacterial type diseases. T are cell-mediated immunity. They have to get next to the cell they're destroying. B produces antibodies which activate complement. They can reach out and destroy from a different distance. B is humoral immunity. T is cellular immunity. It's cell-mediated. That's T. B is humoral or antibody. Secondary lymph organs are like the spleen, the tonsils, and the lymph nodes. The lymphs get there. They either they multiply. Some are memory cells. Some make antibodies. Or in T cells, they go out searching for the pathogen. Local inflammation. You get bacteria or something under the skin. This is where you get the swelling, the redness, the heat, the pain, sometimes the pus. Again, generally it's local inflammation is a B. Unless it's allergic, and then it's histamine, then it's IgE. If you get rid of IgE, you get rid of the allergic reactions. Mast cells secrete heparin. Let the blood get there. Again, local inflammation, warm, swelling, pain, redness.
In response,
B lymphocytes, like we say, make the antibodies, humoral immunity. They have memory cells, so once there's exposure, they can start making antibodies in as little as 2 hours on the secondary exposure. Primary exposure may take a week or 10 days. Antibodies, IgE, IgA, IgG is the main one. It binds complement. IgA is the one that's passive, passed on. IgE is allergic. IgM is the primary, or the first one you see, and IgG is kind of unknown. You have a few trillion types of antibodies. They're not all genetically made. You have to be exposed to some of them.
to have separate
Complement, I'll let you look at. There's a few questions on it. T lymphocytes are cell-mediated immunity. They've got to get close. They're cytotoxic. They destroy bodies, generally with virus, fungus, or cancer. They secrete preferins or granzymes to trigger the apoptosis. They break the cell membrane. Helper T improves, takes the antigens, presents them to the B and T cells. Lymphokines are like cytokines to lymphocytes. T cells are receptor proteins. Again, they take the antigens and present them to the lymphocytes. Dendritic cells do the same thing. Histocompatibility, tissue is tissue compatibility. Know what an MHC is, major histocompatibility complex. Know that they present foreign antigens to both T and C and B cells.
processed differently. And synthesis.
Okay, T cells, macrophages secrete interleukin-1. T cells secrete macrophage colony stimulating gamma interferon. They help activate the cytotoxic T cells.
Require the
At the end of the infection, the T cells are destroyed, and there's an FAS protein or FAS ligand that keeps immunologically privileged sites like the testes and eyes free of T cells. Active immunity is exposure like a virus. Passive immunity is what's passed on from your mother, things like that. Tumors differentiate to an embryonic state, and that's how we can tell them. They're not as differentiated as an adult cell or a regular cell. We have natural killer cells, recognized malignant cells, and other infected cells such as viruses. We have autoimmune diseases where the body's attacking itself, arthritis, diabetes, multiple sclerosis, lupus, where the body, in lupus, where the body attacks its own DNA. Allergies, hypersensitivity, generally it's IgE. You have immediate, which responds very quickly. Delayed may take weeks, or days, excuse me. Immediate would be like an allergy to penicillin. Delayed would be like an allergy to poison oak.
Some people develop.
And that's the end for 15.